So today we will discuss interphase. We already have discussed interphase, the phases of interphase, because in cell cycle we know that there are two phases. Cell cycle completes in two phases. The one is interphase. The second one is mitotic phase or M phase. After that, we have discussed interphase in detail. That interphase completes in three phases: G1 phase, S phase, and G2 phase. But I have some questions from you. If a cell have mutated DNA, if there is some mutation, there is some change in normal DNA, can the cell divide? Can the body give permission for division? Why? Because in interphase we have discussed 2 points, and that 2 points were called checkpoints. Checkpoints. For example, if we have a parent cell here, this is the parent cell, and this cell is now planning to divide. Because what is interphase? Interphase is actually the preparation for division. It is a phase in which a cell prepares itself for division, right? So this cell is now planning to divide. But on the basis of cell division, we have three types of cell. We have discussed that in detail. But one is labile cell, the second is stable cell, and third one is permanent cell. What is labile cell? Continuously dividing. What is stable cell? They divide when only needed. What are permanent cell? Once they divide, they cannot again follow this pathway, are called permanent cell. So we have three types of cell on the basis of cell cycle or cell division. One are labile cell, labile cells. Labile cells examples are cell of GI tract and skin cells. There are a lot of endothelium, vascular cells, right? The second one is stable cell. When some stimulus received by stable cell, they will divide. The stable cells are hepatic cells, hepatocytes, and kidney, renal cells, nephrons. Nephrons are stable cells, right? The third one is permanent cells. Permanent cells. Permanent cells cannot divide. Once they divide, in fetus stages, they will divide and develop other cells for completing the organs, for growth. But once they complete it, they cannot divide it again. Even if they are injured, if they are dysfunctioned, they cannot, they have the ability to divide. And they are neurons and cardiac cells. In cardiac, the specific cells are myocardium, the middle layer, right? So now this cell, for example, this is labile cell or stable cell, and he is planning to divide. It will pass from three phases. First, how much time taken by interphase? 12 to 18 hours. 6 to 8 hours only take by G1 phase. G1 phase. So this is G1 phase. In G1 phase, we have discussed that cell got some changes. The first change is protein synthesis. The second one, hormone cell grow in size. Cell grow in size. But we will focus on this protein synthesis, right? For example, if we have a car and we are driving, we have two things. The one is accelerator and the second one is brake. By accelerator we are speeding the car, and by brake we are stopping the car. There are two things, molecular level. One is molecular accelerator and the second one is molecular brakes. And they have also two brakes. Like in car there are two. One is emergency brake, which is called hand brake, and the second is normal. There are also two brakes. One is working in emergency condition and one in normal condition, right? So when this cell grow in size, protein synthesis enhanced, it will jump into S phase. And S phase is specific for DNA replication. The point which I want to mention here, that there is a point between G1 and S phase where this cell is checking by some proteins or some other mediators, right? And if this cell have any mutation, a small mutation, that this cell is not allowed to go to S phase before they repair their DNA or cell, right? There are a lot of proteins responsible for this repairing, for this screening, for the inhibition, for activation, right? There are some receptors, genes, right? So this checkpoint is called G1S checkpoint. We will discuss today G1 phase, G1S checkpoint, and the next thing is G2 checkpoint. There is one other checkpoint here after S phase. When DNA replicates, one checkpoint is here, which allows this cell to G2 phase, because after this G1 phase and S phase, the cell is now how many chromosome? 4n, or it have 2n amount of DNA, right? Called arrested phase, right? So now one another thing, what is mitosis actually is? What is the difference between M phase and cytokinesis? Anyone? Hmm? What is mitosis? Division of cell. Division of cytokinesis. Cytokinesis is division of cytoplasm, and cell divides when cytoplasm is divided. Anyone from you? Only the nuclear division is called mitosis. Only the nucleus is divided, because in these phases before cytokinesis, in telophase, you see that cell having two nucleuses. The cell having two nucleuses, having 46 chromosome and 46 chromosome. This division of cytoplasm take place in cytokinesis. So if someone asks from you that what is mitosis actually is, or what is the actual difference between, what is the real difference between mitosis and cytokinesis? Mitosis is just the division of nucleus, and cytokinesis is the division of cytoplasm. And when cytoplasm divides, it divides the cell organelles equally into daughter cells. So that is also the function of cytokinesis, the equal distribution of cell organelles. Mitosis is only responsible for this nuclear division, right? So we will discuss today this in detail. For this, we will take this cell and draw it big here, and we'll see that what are the changes occurs in G1 phase. We will, yes sir. Now, for example, this is G1 phase, this is S phase. In G2 phase, the repair of this cell occur again. If there is some fault, it will removed by some of the mediators present in cell. After this, we have M phase, and M phase completes only in 1 hour. This interphase takes 18 hours or 12 hours, 16, 15, 14 hours, from 12 to 18 hours. But this M phase takes only 1 hour, M phase. And M phase is completes in prime minister T, prophase, metaphase, anaphase, telophase. And the cytokinesis is the last step, which is the part of telophase, or which is separate process in which the cell organelle is equally distributed in cell, and the cytoplasm divides by actin and myosin. Right? Now, when this cell divides, for example, after M phase, these two daughter cells formed. Daughter cell. If these cells are labile, if they are labile, they will again start this cycle. If they are stable cell, if they are stable cell, then these cells will go to G0 phase or G0 phase. And G0 phase is resting phase. This is the resting phase for cell, because in this the cell cannot divide. It is waiting for a stimulus. For example, if growth factors occurs and tell the cell that this is the time to divide. For example, if we transplant a liver, for example, we have liver. When the weight of this liver completed to or become 1.5 kilogram, it will stop division. Right? So all the hepatocytes are in G0 phase or G0 phase. But if you cut this area of liver and transplant it to other person, now the growth factors for these hepatocyte will formed in body, and we tell these G0 phase cells, these are hepatocytes which are in G0 phase, that now this is the time to divide and liver start division. When the weight again become 1.5 kilogram, it will stop division again. Right? And here also the mediators formed in this body, and this liver will also complete. Right? So this is stable cell. But in permanent case, in the case of permanent cell, when this cycle completes once, all the cells goes to G0 phase, and they can never then goes to this phase. Right? No stimulus is present for these impermanent. Right? Now we will discuss this G1 phase in detail, and this topic will also cover the regulation of mitosis or the regulation of cell cycle. Right? Now we will take this cell on board and we'll see that what are happening in G1 phase before replication of DNA. What events occurs in this cell? We will discuss 2 or 3 pathways here. The first pathway will be the TK, RAS, RAF, MEF kinase pathway. The second one will be TK, JAK state pathway. Right? And in last we will discuss the P53, P21, 27 pathway. Right? And these all are responsible for this G1 phase. Right? So we'll start now. Pare pushwe? Yes, sir. Question studies pure. Concept ko taso warzai no khair de warshi. Kasa karumi. Di sir taso khair de sir taso dita kasa comment ka no. You are the best. और देना बस बता तुझे वाई तो कुछ इधर जंबार और तो कुछ फॉरेन कंट्री की बता सकते हो वाई Interphase completes in three steps or three phases. We have discussed, right? What are these phases? This is important, right? I am focusing on interphase again and again because there are a lot of cancers type which are related to interphase. This is interphase which is responsible for cancer, right? If there is abnormalities occurs in proteins and mediators and receptors of interphase, it will cause different types of cancers, right? And as a medical student or as a doctor, as compared to mitosis, it is important to know what are the causes of cancer in interphase, right? Mitosis is not important. That causes are important because you are going to treat that causes for preventing or for treating the abnormalities occur in body. So interphase completes in three phases: G1 phase, S phase, and G2 phase. Right? From where interphase occurs, this is cell cycle. Cell cycle. One step is interphase and the second one is M phase. M phase is very simple and interphase is too much complicated and it is too much regulatory phase. The body is trying to regulate, the body is trying to avoid all the mutations or changes in interphase to convert this OK cells to daughter cells, right? If there is some mutations or some changes, mutation mean changes in DNA, right? So, for example, if papilloma virus, if papilloma virus occurs and cause some mutations in the proteins of interphase, it will cause cancer in cervix, right? This is the main cause of cancer in cervix, papilloma virus. We will discuss just in few minutes, right? In G1 phase, simply we said that protein synthesis occurs or enhanced in G1 phase. The second, cell grow in size, right? But remember, I have already tell you, for example, if we have parent cell and it have a DNA, and now this cell is planning to divide, right? It will pass from G1 phase. This is, for example, G1 phase, right? And will enter into which phase? S phase. In S phase, DNA replication occurs, right? DNA replication will start, but before the cell really jump into DNA replication, there is some point which is called checkpoint. Checkpoint. There is some grandfather of genes in DNA, right? And he is ordering. He is very angry man, right? So in this checkpoint, that grandfather will works, right? And he will allow the cell to S phase. If he is not allowing, if he is not giving permission to cell, the cell cannot go to S phase, right? So we will take this cell now. This parent cell, this is parent cell in G1 phase. What are the changes occur in G1 phase? We will discuss this in detail now. For example, I have this parent cell here. This is the nucleus of this is our parent cell, and now this cell is planning to pass from G1 phase, right? What will be the changes occur in G1 phase? We will discuss this in detail now. Right? There are a lot of things you are going to learn today. Right? And how you will compare the bitterness of your today with yesterday. When you are learning new things, you are better, right? And there are a lot of new things for you to learn, right? We have DNA, right? We have DNA here, but I have already told you that in DNA, for example, if I have this chromosome, and this chromosome, I have paternal DNA, I have paternal DNA, and I also have maternal DNA, right? So if we have DNA here, they will have two parts. One is paternal strand and the second one is maternal strand, right? So for example, this is that DNA. This is the DNA present in cell. For example, this is the other strand of right, and this whole DNA is composed of genes. These are genes. Genes are present in the form of codons. This is double helix structure. And who discovered this DNA? Watson and Crick in 1944. Good. You are also got interest or attraction to biochemistry like Liebig, right? And I hope you will do better, right? So this is the DNA present in cell nucleus, right? And this cell is going to pass from G1 phase and G1 is checkpoint, right? What will be the changes occur inside this cell? Imagine that car which you are driving, a car which have an accelerator and brake, right? This DNA also have one accelerator by which he or it accelerate the cell division, and it have a molecular brake by which it stops the cell division. For example, I will tell you names later, but here I only trying to understand you or learn you this mechanism, simple mechanism. For example, if I have a gene here, this is the gene, and when this gene is activated, the cell is replicating or the cell is dividing, and when this gene is prohibited, it will stop the division, right? For this gene here, some brakes and some accelerators are present on this DNA. But remember one thing lifetime in your mind, that we have two strands in DNA. For example, this is one strand and this is another strand, right? This is maternal and this is paternal. If maternal strand have gene here for cell division, on the same location in paternal it also have a gene for cell division, right? If there is a gene for stopping cell division, the paternal chromosome must have the gene on same location for stopping of cell division, right? So if mutation occur, if some dysfunctionality occur in one strand, the chromosome is still able to perform that function, because the maternal, for example, this is dysfunctional, this gene is dysfunctional, this gene is working, right, and this is enough to control the activities. But if both the genes will get some mutation, it will affect now the cell division, right? So for example, we have accelerators here, and these are the, for example, this person are trying to accelerate this gene, and when it activate this gene, the cell division will starts. And we have another, this is the grandfather, but he is very fond of sleeping. He is sleeping every time. This gene is sleeping every time. He only awake when needed. When someone is working wrong in cell, he is activated by someone and disturb him that awake. Some peoples or some persons are working wrong in your area, right? So he become angry and stops cell division, right? So we will discuss that pathways for activation of these genes and for inhibition of these genes, right? So let's start from that points, right? We have specific genes for cell division. The first gene, which is for example present here, which is present here, for example this gene, the point which I have discussed of paternal and maternal is like this. For example, this is maternal strand and this is paternal strand, right? I have a gene present here. This is the gene, right? This gene present on maternal chromosome and paternal chromosome also, and this is called alleles or homologous chromosomes, right? This gene is called growth factor gene. This gene is called growth factor gene. Growth factor gene, right? What this gene is doing. For example, this is stable cell. For example, this is stable cell, and it gets some signals from brain for cell division, right? For example, if we transplant a liver from one person to another, the brain activated and signals this gene and activate this gene, and what this gene is doing, this gene produce a factor which is called growth factor, right? This is very simple. Growth factor gene produce growth factor. What is the process by which it will form growth factor? Protein synthesis. And we have studied that G1 phase is responsible for protein synthesis, and the growth factor is protein, and this protein will be found by protein synthesis, transcription and translation, right? So it produce a factor. For example, this is that factor, right? This is called growth factor. Growth factor, right? This growth factor comes to cytoplasm. Here is nuclear pores. Do you know? This is nuclear membrane. This is nuclear membrane. So here is nuclear pore. and by nuclear pore this protein comes out from first messenger RNA will formed, and then that messenger RNA will comes out and will produce this growth factor protein from ribosome. But we are not discussing protein synthesis, we are just discussing G1 phase. If someone is really wants to learn this, when growth factor gene is activated, when growth factor gene is activated, this area of DNA will expose as express, the transcription will starts, and it will form messenger RNA for growth factor protein, that will called growth factor protein messenger RNA. This step is called transcription. It is called transcription because this is DNA. Growth factor gene is present in DNA. So from DNA messenger RNA is formed, and this messenger RNA will comes to cytoplasm. To which part? Ribosome. In ribosome, it will order the ribosome to form the growth factor protein, and the ribosome will form growth factor protein, right? And this is the growth factor protein. It comes out here, growth factor protein. Now he is waiting that why I am here. When we got some accidents and will unconscious and suddenly awake and we are in hospital, we are just surprised, where am I, right? This protein is surprising what I am doing in cytoplasm, right? So in neighbor of this growth factor gene, there is another gene present here. This is another gene present here. It will activate this gene. What is the name of this gene? Anyone? This gene is called growth factor receptor gene, right? Growth factor receptor gene. It will also activate it and express this area of DNA and will form the messenger RNA for growth factor receptor. Now recap your cell membrane. What receptor is? Receptors are proteins, right? Receptors are extrinsic proteins, sorry, right? So for that protein messenger RNA formed, and this messenger RNA comes here. But I have told you that our external nuclear membrane also have ribosomes, right? External nuclear membrane also have ribosome, and that ribosomes are activated now and forms a receptor which is called growth factor receptor. This is the growth factor receptor, and this receptor have two parts. One is extracellular domain and the second one is intracellular domain. One is here the extracellular domain and here is intracellular domain, right? What is the name of this receptor? This growth factor receptor, commonly it is growth factor receptor, right? But what is this protein called? This is called tyrosine kinase, right? Abbreviated by TK, right? Tyrosine kinase. What this tyrosine kinase is doing? He opens his mouth and till this growth factor proteins, growth factor protein come here, right? And this growth factor proteins binds growth factor receptor. Growth factor protein will bind to growth factor receptor. Now this receptor is activated. This is activated. I will tell you that what is the mechanism behind this activation, but for now this receptor activated, right? Now he is asking why I am activated here, for what purpose, right? So for that question, the answer of that question is given by another gene here, right? And that gene is called, for example, we have a gene here. This is the gene, right? He is hanging here. Huh. تاسو به دې باندې پوهیږئ که نه هسې راته نه پښتو کې به دا اله وایم چې کله دا پوره کړم که داسې ورته باندې منې مه کوم کنفیوز به یې ټول صحیح ده. So this is another gene and the name of this gene is called, the name of this gene is, any one of you signal transducer gene, right? Signal transducer gene, and this gene signals, this gene will get signals from here because it will form a protein and that protein is present. For example, that protein is called signal transducer proteins, right? Signal transducer proteins, and these are, for example, signal transducer proteins. Signal transducer proteins. Signal transducer proteins will bind to this receptor after activation, and this receptor is activated by growth factor protein, which are formed by growth factor gene, right? So this is now activated, and this he ran here and bind to growth factor receptor intracellular domain, because this is called extracellular domain and this is intracellular domain, right? And now he is signaling, he start signaling. All these signals are directing outward, right? This growth factor complex gives signals in this direction, right? But what are these signals? I am telling you in just a few seconds, right? This is called signal transducing protein because it giving signals to genes. Right? So it will give signals to another gene, and this gene is called responder gene. The gene is called responder gene. This is responder gene. This is responder gene. This is called responder gene. These all genes are working right now in my body, right? This is responder gene. What responder gene will do? It will now start transcription and will form transcription factors. Transcription factors. These responder genes are activated by signal transducers proteins. These are signal transducer proteins. All the proteins are formed by that mechanism, the transcription and translation, right? So these signals are receiving by this responder gene, and this responder gene will respond in the form of formation of a protein called transcription factors. For example, these are transcription factor, right? These are transcription factor, and now these transcription factors are responsible for activation of this cell division by a mechanism which is called cyclin dependent kinase mechanism, right? Cyclin dependent kinase mechanism. For that we have a proteins, we have a gene here, right? Because this is called genetics. This is called genetics. This mitosis and meiosis are the processes of biochemistry actually, right? So biochemically it is regulated like this, right? So we have a gene here. For example, this is a gene. It is cycling, right? And this gene is called cyclin protein gene. Yeah, cyclin synthesis gene, cyclin synthetic gene, right? This is activated by this transcription factor. This is called transcription factor, right? This cyclin protein is gene is activated, and this area of DNA is now expressing mean it will now form proteins. For protein synthesis start here. This is stepwise processes. First protein synthesis occur here, then here. These are the genes, or these are the codes, right? And ribosome is responsible for making the proteins according to the message of DNA. So here the DNA message is formed growth factor protein. Here the DNA is messaging to form growth factor receptor proteins. Here the DNA is messaging to form the signal transducer proteins, right? And here this area, this DNA is messaging for formation of transcription factors. And these transcription factors activate this area, and this area will message the ribosome to form the cyclin proteins. Cyclin proteins, right? These are cycles of proteins. These are actually the cycles of another protein. They are cycling, they are riding on this cycle, right? But when the cycle is present, the protein can work. When the cycle is absent, he cannot work. So they are called cyclin dependent kinases because they are dependent on cyclin proteins, right? For example, this is a cycle. This is a cycle, cyclin protein, right? If the cycle is present, the protein will work. If this cycle is not present, this protein will not work. And what are the genes called? These are that gene, and this gene is called CDK, mean cyclin dependent kinase gene, right? What is kinase? What are kinases? Anyone? What is kinase actually say? The kinase are the enzymes which are responsible for phosphorylation, right? Because during glycolysis, if you think the hexokinases, fructokinases, they are responsible for phosphorylation. So these kinases are also responsible for phosphorylation, and this phosphorylation is responsible for all these functions, right? So this gene is activated, this gene, right? But remember one thing, for lifetime in your this transformer, that this gene activated every time, it is working every time, and every time it get ready to ride on a bicycle and goes to divide the cell, right? But this cycle is not present every time, right? And this is cycline dependent. So if this cycline is present, he will work, and if this cycline is not present, he will not work. And this cycline is formed by this pathway, signal transduction proteins, and so mean that if there is no signal transduction proteins, cycline will cannot formed, right? And this cycline dependent will not work, right? So the protein formed by these cycline dependent kinases are, for example, this one is cycline dependent protein, cycline dependent kinase proteins, right? When he is formed and see the cycle, he run and ride on bicycle. So this protein come here, right, and sit on this cycle, right? This thing is now called, this protein is called cycline dependent kinase complex, right? Cycline dependent kinase complex. And what he is doing, what he is doing, I already told you that there are some breaks also here which stops the protein. Now leave this pathway for a few seconds, right? Because we have protein here which hold the cell division, which hold the key for the key of the door which is responsible for cell division, right? Because there is one door, and behind that door this gene is present which is called DNA replication gene, DNA replication gene. And if this gene is activated, the DNA will start S phase, because DNA replication occurs in S phase. So if this gene is not activated, this cell cannot go to S phase, right? There are a lot of barriers. There is a door in front of this gene. Also there is a dog present here. A small dog is sitting here and hold the key in mouth, which is the key of this door, the door which is going to this gene, right? So we will talk about that dog and that key, right? Or crocodile, because this is very dangerous. So a crocodile is sitting here, right, and eat all the things which comes here, right? But there is one thing which phosphorylate that protein and take the key from that protein, right? So we have a protein present, a gene present here which is called RB gene, and this gene is responsible for holding this cell from cell division, right? What is RB stands for? Retinoblastoma gene. Because when this RB gene discovered, the patient were caused by retinoblastoma. This is a disease of eye, right? So this protein, these proteins are present in their cells, and they call them RB genes, right? This RB gene is responsible for making a protein which is a crocodile sitting here. I don't know how crocodile looks like. right? He is sitting in front of that door, right? And holding the key in his mouth. This is the key for transcription or activation of this gene, right? No one is able to take this key from this. And for the time when the key is in the mouth of this crocodile, the cell cannot divide because this gene is not expressing, right? And this gene is responsible for DNA replication, right? Now come here to this CDK complex kinases. These are kinases enzymes. So it will hit the crocodile on the area where it hurts, right? So this cyclin dependent kinase occurs here and phosphorylate this protein. What is the name of this protein? This crocodile. What is the name of this crocodile? RB proteins. RB proteins, because they are formed by RB genes. So these are RB proteins, retinoblastoma proteins. And when a cell have retinoblastoma protein, the cell cannot divide, and we called it stable cell, right? Stable cells have this protein, and this protein is holding the cell from division. It prevents the cell division, right? But when needed, this pathway is activated, and this cyclin dependent kinase complex occur and phosphorylate this. So the crocodile start jumping, right? There, because he is disturbing. When he is start jumping, he opens his mouth for shouting, right? When he opens his mouth, this key goes here, right? And binds to DNA replication gene. And by activation this gene, this DNA will start replication, which is S phase, and we will discuss that in detail, right? But what is that checkpoint? Where the checkpoint is now DNA is replicating and checkpoint is hidden, right? The genes which are responsible for that checkpoint are very important, because here are in the neighbor of these RB genes. There is one gene which is called, let's talk about that grand gene. This grand gene, right? Which is the father of all the genes during replication, right? And he is very serious. He is very serious to his work also, and to the work of others also. He is taking all the work seriously, right? He is not joking. And the work of this gene is sleep. He is sleeping. And tell all the gene, do not disturb me. Don't disturb me, right? He is sleeping. This gene is sleeping, right? And he is sleeping in a bed, right? And that bed is called MDM gene. So this is MDM gene, right? And this is very important, this red one, and this is called P53 gene, right? If a questions asking in paper that what is the grand genome, the option will be P53. P53 is the grand genome of all the genes, right? Now he is sleeping in a bed of MDM, murine double minute 2 gene. This is called murine double minute 2 gene. Right? Now these are unactivated because the cell is going to divide. Right? Here is a police and checkpoint, because this is, we are talking about not checkpoint, this is the checkpoint. For example, if we have interface here, this is cell cycle, this is full cell cycle, right? This is G1 phase, right? And this is S phase. This is the checkpoint, right? If the cell have any mutation, he will be arrested here, and for arresting, a police is present here, and the police is called ATM, right? Police is standing here, this is the police. Why you are talking? Why you are talking? Any problem? Then focus here, right? This is the police standing here, and this police also have a telescope, right? Because he is in a very keen observation. If someone working wrong here, he direct report to grandfather, right? So he has a telescope, right? And every time he is scanning DNA, he is scanning DNA from here up to here, from here up to here, he is seeing to this DNA. There is some mutation in the DNA, some change in DNA, or the DNA is okay. If the DNA is okay, he allows this cell to pass through this checkpost and goes to S phase. But if, for example, this is the DNA, and this DNA have some mutation here, right? He absorbed this uh, DNA mutation, right? What is the name of this gene? ATM, or it is called DNA scanning gene. DNA scanning gene. What does ATM stand for? Automatic? Uh, Ataxia telangiectasia mutated gene. ठीक है? It is called ataxia telangiectasia gene, right? This gene is responsible for scanning the DNA again and again, right? So if he observed some mutation in DNA, he will direct report this P53, right? And P53 got angry. Why you are disturbing me, right? And awaked. And the activation of this P53 is he leave this bed, MDM, right? When he is unattached from this MDM, he become activated, right? So he call this P53 that there is some mutation, DNA is not working properly, right? Awake in your presence, DNA is not working properly, right? So he get angry, right? He get angry, right? This is now activated P53 gene, right? What is the role of guardian in home? if your car gets some accident, first he release his angerness on you, why you accident your car, right? And then call the mechanic, come here and repair this car, right? Like that, he first become angry, right? And then activate a chain of genes here, right? One is called one is called P21, right? Second is called P27, right? And P25, P24, right? These genes goes here because this is now the order of this P53, the grandfather, right? And these both the proteins make P27 protein and P21 protein. They goes and hold this whole process, stop this CDK complex to go to phosphorylate this protein, right? RB protein are now holding by these two P21 and P27, right? And the cell stops division. This is the checkpoint, this is the arresting of this cell, right? Now he call another gene here, which is called the DNA repair gene, the DNA repairing gene, right? He is in his shop, right, and having a lot of instruments for repairing, right, a lot of proteins, a lot of other things, right. He came where to this mutated DNA and remove this mutated gene from DNA and repair the gene DNA, right. When this DNA got repaired, this grandfather P53 comes back to his bed, right, and sleep and order that work properly, right. And now the cell again start DNA replication, right. Now this is the story of how this checkpoint works, right. But if there is mutation in DNA which is more than that, these proteins are not able now to repair, right. This ATM orders again, disturbing again and again this protein because he got some mutation. This pathway follows again. When there is mutation, he again order to this P53. So P53 is working again and again and again and again, right. So he got a lot of angerness and tell the cell, stop this division, stop this work. He acts on these genes, right, order these genes to stop this pathway, right, and activate one thing which is called apoptotic gene, which is called apoptotic gene. For example, this is that gene, apoptotic gene, apoptotic gene, right, and actually it is called pro-apoptotic gene. When, for example, we have DNA and it got greater mutations, right, and now this protein is not able to repair this, right. So this grandfather orders this apoptotic gene to be activates, right, and when this gene activated, it will finished or it will diminish this all pathways, right, and the cell will now not able to divide. If they are stable cell, even then they are working, the cell is working, but having no ability to divide, right? These are the cells, right, which are not now able to divide but work properly, right? These genes are called, collectively these genes are called proton kick gene, right? Proton को जीन है, proton को genes. Collectively, these are called proton को gene. When they are working properly, when they are working properly. But now come, when there is some mutations occur in these genes, right, and they are activated by itself, it will enhance the cell division, because it will phosphorylating, it will follow the transcription translation and will follow this pathway again and again and again, right? For example, if we have the cells of stomach and got some mutations, right, got some mutation, these cell will start abnormal divisions, right, and this is called cancer. This is called cancer. The virus which I mentioned earlier, the papilloma virus is responsible for phosphorylating of this crocodile gene, this crocodile protein, right, abnormally. When papilloma, for example, this is papilloma virus, it comes and activate this. Now there is nothing to hold this key. What is the name of this key? Anyone? What is the name of this key? It is called E2F transcription factor. E2F transcription factor, right? Now, if papilloma virus infect this cell, basically papilloma virus infect cervix cell and caused cervix cancers, right? So these papilloma virus regularly enhance this phosphorylation, and this phosphorylation, now there is nothing to hold this key, right, and the cell, there is no signals to stop the cell division, and abnormal cell division starts. The cell is dividing again and again and again, and it forms tumor. For this reason, this RB gene and this P53 gene, P21 and 27 are called tumor suppressor genes. They are called because this is called tumor. This is called tumor, right? Rasole, right? So these genes are responsible for stopping the cell division. So they are called tumor suppressor genes, right? Now again come to the receptors, because we are now will learn the receptors, right? This signal transduction proteins, this protein, it will activate a protein because these are signaling, and signaling is, for example, if I say to CR that go and tell Hizran Sahib to come here, right? So there are two persons, right? This signal transducer proteins tells the protein which is called RAS proteins, called RAS protein, that tell the responder gene to respond against cyclin protein, right? This RAS protein tells to another protein which is called RAF protein, right? This RAF protein orders another protein which is called MAP kinase. Mitogen activating protein. What are mitogens? The growth factors. The growth factors are called mitogens because it activates mitosis. Mitogens. The growth factors may be growth factors. These growth factors may be epidermal growth factors. They are also called endothelial growth factors. They are also, they can be or may be vascular growth factors. The area which is where it enhance the mitosis, right? They get their names, right? Epidermal growth factors, endothelial growth factors, vascular growth factors, etc. And collectively, these growth factors are called mitogens. Mitogens. Mitogens are the substances which enhance the mitosis, right? So this protein is called mitogen activating proteins. Mitogen activating protein. Mitogen activating proteins, right? And it will follow this pathway, right? But I have discussed in cell membrane that there are some types of receptor, 1 pass receptor, 7 pass receptor, channels, right? This is actually work on 1 pass receptor. It can be work on GS and GQ or 7 pass receptors, right? And these 1 pass receptors are called tyrosine kinase, right? This is normal pathway, normal when growth factors occur. But here leukotrienes also take parts, leukotrienes, right, which are the swelling mediators, right? So the receptors which are present for other growth factors, for example, this is 1 pass receptor, this is 1 pass receptor, right? And the growth factors came here and bind to this, right? These proteins become activated, and this, what is this? Signal transducing proteins. So signal transducing proteins comes and binds here, right? When they are activating this couple of receptors. The mechanism behind this is they are far away from each other. These two receptors. But when they are activated, they come close and hug each other, right? When they are joining, this protein came and binds here, and in this case, when these two receptors are activated, it will activate a protein called JAK proteins, genes kinase proteins, right? And these genes kinase proteins will activate state proteins. State stands for signals. This state is called signal transducing. Mata sa kabur wili. Samkha. Signal transducens and activator of transcription, right? These state proteins give signals to this responder gene. So this pathway is called TK RAS RAF MAP kinase pathway, right? And this pathway is called TK JAK state pathway, right? These are the two pathways which are working behind this mechanism, right? There are two mechanisms remaining. We will discuss that in detail after a short break, right? Yesterday we have covered the G1 phase in detail, but only the two things which are remaining are GS receptor activation and GQ receptor activation. There is a hide mechanism behind these two, and we will discuss that mechanism, like growth factor or growth factor receptors or TK, RAS pathways, like that it will also activate some pathways, and by that pathways it will control the cell cycle, or it will control the parent cell to pass from checkpoint to S phase, right? So we all know that interface completes in three phases: G1 phase, S phase, and G2 phase, right? But there are two checkpoints where the DNA or the genes checking the cell, the parent cell, that is this cell okay to enter into a space or complete this cycle or not, right? And that points are called G1 is checkpoint, and the second one is G2 checkpoint, right? For example, we have a full cell cycle, and full cell cycle completes in two steps. One is interphase, this is interphase, and this one is M phase, right? So in interphase, this cell is trying to prepare for M phase, and in G1 phase, the cell is preparing to enter into S phase, and in S phase, it will be prepared to enter into G2 phase. If there is any mutation, or change occur in DNA or in other components of cell, the checkpoints or the enzymes which we have studied cannot allow that, not giving the permission to this parent cell to enter into next phase, right? This is G1 phase, and this is that checkpoint. G1 is checkpoint. Which one is the master gene in this checkpoint? P53. P53, because this gene controlling the activity of G1 phase, right? And G1 phase, the most important thing which taking place is protein synthesis and cell growth, right? For example, this is parent cell. This parent cell is passing from G1 phase and trying to enter S phase in DNA replication, right? Here we have studied some events in this parent cell: the function of growth factor, the function of P53, the function of RB proteins, right? Cyclin dependent kinases, cyclins, right? Now we will study the weight mechanism which are controlling by other receptor, by 7-pass or other receptors. 7-pass are complex receptors, G protein coupled receptor. 7-pass receptors are called G protein coupled receptor. So we will take this cell again here. For example, this is that parent. This is that parent cell, and parent cell have 2 nDNA or 1 nDNA, 1 nDNA, 1 capital nDNA, and having 2 n number of chromosomes, small n, right? So we will take that DNA here. This is, and each chromosome have one original strand DNA and one copied strain, like, right? And in S phase we will discuss that how these copied of these strands are making by this parent cell. This is the activity of S phase. But now we are only preparing this cell to enter for S phase or DNA replication, right? So we have DNA having original and copied strand, and having 46 number of chromosome, mean 23 is from parent father and 23 from mother, right? For example, this is the second strand of DNA. What is gene? Gene is the encoding information on DNA. The code which store the information on DNA is called gene, right? But in father and mother chromosomes, the gene location will be the same, right? So, for example, these are the DNA molecules, and we have genes. Tell the name of this gene. Growth factor. Growth factor gene. Tell the name of this one. Growth factor receptor. Growth factor receptor gene. Right? Tell the name of this one. Signal transducers gene. Right? Tell the name of this one. Responding gene. This is responding gene. Responder gene. Right? And these are the accelerators of cell cycles. Accelerators of cell cycle. Now, tell the name of these guys. RB protein. This is RB protein. RB stand for retinoblastoma. Gene. Protein. Not protein. Sorry. Gene. Right? What is the name of this grandfather? This is P53. Sleeping in MDM. MDM. What is the name of this gene which are scanning? He has a telescope. Right? That police. ATM. Ataxia telangiectasia gene. Right? Next one. This gene is repair gene. DNA repair gene. Right? The next one. Tell the name of this one. P21. Tell the name of this one. P27. P21 and P27. Alright? These are the breaks of cell cycle. These are the accelerators of cell cycle. Molecular accelerators, molecular breaks. But this one, RB, P21 and P27 are foot brakes. And this one is hand brake. This is emergency brake. Right? Here is one gene which is very, very important. Not cyclin. The DNA replication gene. DNA replication gene. This is just the recap of yesterday lecture, right? The next which is very important in the form of a cycle. Here, this gene is cycline synthesis gene. Not, these are not protein, these are genes. According to these genes, proteins will be formed in G1 phase, because in G1 phase we are going from protein synthesis, and protein synthesized on the message or according to the message of these genes, right? There is one crocodile which sitting here, and this protein is called RB protein, and RB protein is synthesized by RB gene, right? But the key for this gene is in the mouth of RB protein, which is called E2F transcription factor. E2F transcription factor, right? When this crocodile leave this key, the cell cycle will starts. These genes are responsible for activation of this protein, right, on a pathways, and these genes are responsible for inhibition of this cycle, right? By this growth factor gene, growth factor protein will formed. Growth factor proteins will form, right? And it follows the pathway. Which one pathway is following? Huh? Biya samegi बार बार समेगी करा की इवा बस खैर देखो सफा यो शान ब्लड होने रखा बिया टिक तब बिया हाँ mambo focus हो बोर्ड फोकस हाँ दादा sini kan dari board mana klik ka pama klik ka apa sa right? So these genes orders for messenger RNA. It makes the messenger RNA for growth factor proteins, and this messenger RNA goes to cytoplasm and forms the growth factor proteins, right? For example, this is growth factor protein. The next one, this, it forms the messenger RNA for growth factor receptors, and it goes and forms the growth factor receptor. We have discussed this in detail. After this protein came and bind to the extracellular domain of this growth factor receptor, right? After this, the signal transducing makes the protein, it forms RNA first for which thing? Signal transducing proteins, and these signal transducing proteins binds to intracellular domain, right? No. Nucleoplasm के अंदर सिर्फ a messenger RNA जुड़ेगी. Only messenger RNA forms a nucleus. It will leave this membrane. This is nuclear membrane. It will leave this nuclear membrane by nuclear pore. And I have told you that to extra or outer nuclear membrane have ribosomes, and these ribosomes are responsible for making this translation step, right? This is transcription step in all the protein synthesis, right? And this is translation step, right? So these proteins formed before coming here, these proteins are formed here, and it again enter into this nucleoplasm, right? This protein also came to nucleoplasm by nuclear pore, because for example, if this is cell membrane, this is cell membrane and this is cytoplasm, This is the outer membrane and having nuclear pores. The messenger RNA leaves this nucleus by this nuclear pore. Here protein synthesized by ribosome, and these ribosome when needed in nucleus, it will goes to nucleus again by these nuclear pores, right? Because nuclear pores are responsible for transport, right? These signal transducing proteins binds here in response of this receptor activation and gives signals by which mechanism? RAS, RAP, MAP pathway, right? It activates a protein called RAS proteins, and RAP proteins are activated by RAF proteins, right? And this RAF protein goes and activate MAP kinase. MAP kinase are responsible for phosphorylation of this responder gene, right? So it gives signals to responder gene, and this pathway is called, this receptor is called tyrosine kinase, and one pass receptor is type of receptor, but the name of this is tyrosine kinase. So this is called TK RAS RAF MAP kinase pathway, right, by which cell cycle is activated or regulated, right? So this receptor responder gene is activated and it orders the cyclin genes to form cyclin protein, and cyclin protein also formed according to, it will form the messenger RNA first for cyclin protein, that will go to the cytoplasm. In ribosome the cyclin proteins will form, and that cyclin proteins will come again to nucleoplasm, right? But it is responsible for formation of one protein which is called cyclin protein. This is cyclin protein, and here one gene which is activated forever, every time the gene is activated, and this is called cyclin dependent. Good, that's mean you are getting interest and learning. So this is cyclin dependent kinase gene, right? This is anytime it is activated and forms a protein called cyclin dependent kinase. Cyclin dependent kinase. But if there is no cyclin, this protein cannot work. If there is cyclin, this will ride on this bicycle and will hit this RB protein by this cycle, right? So when this pathway is activated, it will form cyclin protein, and this cyclin dependent kinases will jump and will set on this cyclin, right, and came here and will phosphorylate this retinoblastoma protein. This retinoblastoma protein is responsible for holding this cycle. For example, if this cell is in G-0 phase, I mean it is not dividing yet, right? So the protein which is responsible for this G-0 phase is RB protein. It holding the key for cell cycle. When this cyclin protein came here and phosphorylate this crocodile, it hit the crocodile on bicycle. Oh, crocodile says, oh, so open his mouth, and when this crocodile open his mouth, this key is now free, and this free ran here and activate this DNA replication gene, and now DNA replication starts, before checkpoint. There are checkpoints, these genes are responsible for that also, right? But this gene is responsible for DNA replication, right? The next thing, if these receptors are, these are also one pass receptor, and for example there are two growth factors, vascular growth factor, endothelial growth factor, hepatic growth factor. They came here and bind to this receptor. These receptors are called tyrosine kinase, are abbreviated by TK, right? When both the receptors are activated, they will join to each other. They are hugging now, right? By this activation, this signal transducing protein will bind to these receptors and will follow another pathway which is called, it will activate the genes kinase proteins which is called JAK proteins. JAK protein, genes kinase, right? It will activate state, and state it stand for signal transducing and activator of transcription, right? This protein will signals responder gene and it will form cyclin and this pathway will followed again, right? These are the mechanisms which we have already discussed yesterday, right? There is one police gene, this ATM, ataxia-telangiectasia gene. All the time it is scanning this DNA. This is DNA, right? It's scanning this DNA on keen eyes every time. If there is some mutational fault occur in this DNA, it is not working properly because there are a lot of proteins which are responsible for this DNA replication, like helicases, polymerases, ligases, right? So if they are not working properly, this police will report to this guardian gene. This is also very important for MCQs, that which gene is called the guardian genome. The P53 is called the guardian genome because it controls all the cell cycle here, right? But this guardian is sleeping in MDM and tell to these all genes that do not disturb me, work properly and don't disturb me, otherwise I will get angry, right? So when this ATM see this mutation, it orders the guardian genome and guardian genome wake up. Wake up mean it will debind from this MDM. It will leave MDM, and when MDM is separated from this P53, it become activated. So if this the ATM reports this gene, it will become activated. It will become activated and gets angry. First it will warn out this ATM and the ligases and polymerases, etc., right? And then will activate P21 and P27. For what? For what purpose? He is telling to these P21, 27, run and hold this cyclin dependent complex. When these both are joining, for example, this is the cyclin protein and this is the cyclin dependent kinase protein, right? This whole thing is called cyclin dependent kinase complex, right? This gene activates P27 P21 and tell run and hold this complex. Hold this complex mean inactivate this complex. When this comes here, it holds this It holds this CDK kinase, and when CDK kinase is inhibited, do you think cell cycle will in progress? No, because this RB is not phosphorylating, right? And this E2F transcription factor is not phosphorylating, not activating. By that, this DNA replication gene will not work. So cell cycle will stop. So it stops the cell cycle for a while, right? And call this gene, this mechanic gene. This is called mechanic gene. DNA replication, DNA repair gene. He is telling to this, come and dimension, finish this mutation in DNA, repair this DNA. He is now running from here and came here. What he will do by scissor protein, scissor protein, right? Scissor protein, it will break this part of DNA, right, and remove these mutating genes from DNA, and will again bind by DNA ligases, because DNA ligase is called the glue of DNA, glue, right? So if this is one time mutation in DNA, and after this repair, when this DNA is repaired DNA, this guardian gene again come here and sleep and tell all the genes works properly, don't disturb me, right? But if this mutation occurs again and again and again, and this P53 is disturbing again and again and again, he will order the apoptotic gene now. He will order the apoptotic gene to finish this whole cycle, right? And then the cell will remain in G0 phase forever, right? These are the old pathways. Now we will go for new one, the 7-pass receptor. The 7-pass receptors are also known as G protein coupled receptors. G protein coupled receptor. Anyone who can tell me, the intelligent one of you, why they are called G protein coupled receptors? The other names for these are 7-pass receptor. The other name for this is serpentine receptor. And serpentine means snake. Serpentine means snake, because the structure of this receptor is like snake. They are 7 times passing from this cell membrane, right? So for example, this is the 7-pass receptor. 1 2 3 4 5 6 7, right? This is the extracellular domain and this is intracellular domain. But to intracellular domain, there are 3 proteins binds, the alpha, beta and gamma, right? For example, one protein is beta, one is gamma, and next one is alpha. But they are in inactivated form. When growth factors occurs and binds here like this receptor will be activated, right? And this G protein coupled receptor have here, they have 3 types, GS, GQ, GI, but in cell cycle only GS and GQ are working. So it have GS and GQ, right? GS is also stimulatory, GQ is also stimulatory, and GI is inhibitory, right? But for inhibition, there are a mechanism working here, so GI is not present. In some research, it is they are saying that GI is also present here, right, but not confirmed. So this is GS receptor, which is 7 pass receptor, right? What is the mechanism behind this? Do you know about GDP? All of you are know ATP, ADP. What is GDP? Guanosine diphosphate. This is just like ATP, right? But when GDP is bind here, this receptor is inactivated, or these proteins are inactivated. When this GDP will convert into GTP, this protein will be activated, right? Here is other protein sitting here and waiting. For what he is waiting? For activation of these receptors, right? This receptor, this protein will be activated by activation of this coupled receptor. So why these are called coupled receptor, coupled protein? Because they are formed of a lot of proteins. This is coupled, right? This is a couple of proteins which are present in alpha, beta, gamma, right? And these are also proteins, so they are called G protein coupled receptors, right? Now, when these growth factors, what is growth factor? Growth factors is also known as growth factors. They are also known as mitogens. Mitogens, right? For example, these are vascular growth factors, these are endothelial, these are renal, hepatic, cardiac, right? They came here and binds to these receptor. From where? From here. This gene is activated. This gene will form growth factor proteins. For example, this is other type of growth factor. Right? This growth factor messenger RNA came here and formed this growth factor, and this growth factors came here and bind to this G protein coupled receptor. When this factor is bind here, this protein become activated, by which thing it will react with one another phosphate and converts into the GTP, right? And when new friends comes, the old are right, right, like that. This alpha protein also leave these two friends, right? When GTP came, these two separated. The beta and gamma are alone now, right? And this alpha converts this GDP to GTP and become like this GTP, right? And now this is activated, right? When GDP is present here, this is called off proteins, off state. When GTP came, it become on, right? What GDP is, alpha protein is doing? Alpha protein goes to this protein, right? This protein is called, anyone? This protein is called adenyl cyclase. AC, these are AC, assistant commissioner, right? And this protein have a big mouth here, right? When this alpha binds to GTP, he is seeing to this, he is watching to this alpha protein, right? And when this GTP came, he is stiff and binds with this alpha and GTP, right? And activated, become activated, right? But he is only using this alpha. The old friends are also gone, and this adenyl cyclase only using this alpha protein by taking his phosphate from GTP, right? So this adenyl cyclase gets this GDP from this phosphate from this GTP, and it become again converts into GDP, right? When the alpha binds to GDP, he leaves this alpha again, right? Because he is only using for money, right? Or for energy. He is getting energy from alpha protein. When this alpha protein become inactivated again, he is now begging to these two again that I have made a mistake on leaving you, right? Their mercy, and tell this alpha protein come here and again bind to this beta and gamma proteins, right? And again become an off state and binds to this receptor, and while waiting for other cycle, right? Other factor will come and will activate this pathway again, right? Now this adenyl cyclase activated. What he is doing? He converts this ATP is present here. All of you know that cytoplasm, nucleoplasm, all the cell is rich in ATP, right? He takes one ATP from here and converts by taking that energy into CAMP, cyclic adenosine monophosphate, right? This is important thing. This is important and called second messenger. This is called second messenger. Second messenger mean this CAMP is second messenger for this pathway. And what he is doing? He follows a pathway which is called phosphokinase A pathway. He activated phosphokinase A protein. Phosphokinase A protein, PKA. Right? And this phosphokinase A protein goes and activates this RAF protein. Right? And this RAF goes to MAF, and this MAF to responder gene. This is the other pathway which is following by cell cycle, right? Which is GS receptor. One is remaining, GQ, which is very simple, not that much complicated, right? So if we have 1, 2, 3, 4, 5, 6, 7. This is GQ proteins, GQ, right? And other growth factor is responsible for activating this receptor. Why these receptors are different from each other? Because I have told you that receptors and enzymes are specific. Only the needed or only the limited substance can activate this receptors and enzyme. So the growth factor which responsible for GQ activations are different from that growth factor which are activating this GS, and the growth factor which are activating this tyrosine kinase are different from these two, right? So if growth factor came here and binds to this receptor, this receptor also have alpha beta and gamma protein, but this alpha is binds to GDP. GDP. When this receptor activated, these two are separated. The alpha and GDP. And this GDP is converted into GTP. So this is now activated. This is now activated. Here to this GQ protein, a protein present in this membrane called phospholipase. Phospholipase. Remember one thing, phospholipase is very important because it converts the phospholipids into arachidonic acids, and by cyclooxygenase 1 and cyclooxygenase 2, that arachidonic acid are converted into prostaglandins, and prostaglandins are responsible for inflammation, fever, headache and body, right? So these NSAIDs, the drugs like Panadol, aspirin, brufen, they are COX inhibitors, cyclooxygenase inhibitors. They inhibit the cyclooxygenase enzyme and blocks the synthesis of prostaglandin, and when prostaglandins are inhibited, the fever, the headache, they are gone, right? But one side effect which is associated with this NSAIDs is gastric ulcer. Why gastric ulcer occur with Panadol, aspirin, brufen, etc.? Why? Because prostaglandin is also responsible for maintenance of the mucous membrane of stomach, and when you are inhibiting prostaglandin, you are also inhibiting that in stomach also, and the acids present in stomach will direct hit with the endothelium of stomach and will cause ulcer, right? So these phospholipases are very important enzymes. They are activated by one other protein which is called PI2, 2PI2 or 2IP2. 2IP2, inositol diaphosphate. It is called inositol diaphosphate. So diaphosphate. Do you remember the phospholipids of upper leaflet and lower leaflet of cell membrane? They are phosphatidyl inositols, and this is inositol diaphosphate. So this alpha activate this protein inositol diaphosphate. It only gets the energy from alpha and GTP and tell the alpha to go back to this receptor for other cycle. This PI2 activates this phospholipase. What is the function of phospholipase? Phospholipase breaks the DAG, diacylglycerol. It is one of the important product of glycolysis. Diacylglycerol, abbreviated by DAG. DAG. Diacylglycerol, right? This phospholipase breaks this into a lot of factors. The two are IP3, or this IP2 breaks this phospholipase into two parts, the DAG and IP3. This IP3 is inositol triphosphate. This IP2 is inositol diaphosphate. Di is for 2, and this is inositol triphosphate, right? This thing is activated, and the other is this DAG. This inositol diaphosphate breaks this phospholipase into DAG and IP3. what they are doing now, this cell have endoplasmic reticulum. Yes, of course, this is cell. A rough endoplasmic reticulum and smooth endoplasmic reticulum are present here, right? So this IP3 is responsible for activation of calmodulin proteins, which is responsible for releasing that calcium. What is the role of smooth endoplasmic reticulum? The storage of calcium. All of you know, right? So this endoplasmic reticulum stores calcium, right? This calmodulin goes and release this calcium from endoplasmic reticulum. So calcium is now present in cytoplasm, and this calcium is responsible for muscles contraction, right? Here this DAG play role. What is that role? That role, phosphokinase C is activated by phosphokinase C. PKC is activated by this diacyl glycerol, and this phosphokinase C goes and activate this RAS protein, phosphorylate this RAS protein, and RAS protein will RAF, and RAF will MAP, and MAP will responder gene, right? So these are the two other pathways which is in cell cycle regulation. After this, if these all things are going okay and there is no mutation, this RB protein and these genes will leave the DNA or the cell to go into S phase. And after this, in next lecture, we will discuss the DNA replication, which will cover two things. One is in physiology, cell cycle. The second one is in biochemistry, nucleic acid, because I am going to teach you the structure of DNA also. And in nucleic acid, the one important topic is the structure of DNA, and we will also discuss the S phase, which is DNA replication, right? These genes are collectively called protoncogenes. But if these, I have told you on clinical point of view that if mutations occur in these proteins, it will cause cancer, it will irregulate this cycle. In that case, when these genes are becoming cancerous, they will cause cellular oncogene. Cellular oncogenes, right? If they are causing cancer, and normal condition they are called protoncogenes, right? The one virus I have discussed you, papilloma virus. When papilloma virus comes here and mutate, enhance the activity of RB proteins, right? This RB protein is not able to hold this key by the effect of this papilloma virus, right? And this cell cycle will taking place abnormally, and this is also cancer. And this papilloma virus is responsible for causing cancer in cervix cancer, papilloma virus. So this is clinical point of view, right? The other things, these genes are called collectively, these are called protonco, and these are called tumor suppressor genes. These are called tumor suppressor genes. Why they are called tumor suppressor? Tumor is something abnormal cell cycle, abnormal mitosis. Like for example, if we say that we have a tumor in stomach, it means this here the cell cycle is going abnormally, right? And it make a mass here. Endoscopy method is used for diagnosis of this tumors or cancers. This is called cancer, right? So these genes are responsible for controlling this cycle and preventing the tumor formation, so they are called tumor suppressor genes, right? By activation of these genes we can treat the cancer, and by inhibiting these genes we can treat cancer, right? So this is all about the G1 phase and interphase, right? The interphase is composed of G1 phase, S phase and G2 phase. We only covered G1 phase, right? And G1 is checkpoint. What is the name of the police on checkpoint? ATM. What is the guardian? P53. What is the normal break for molecular break for cell cycle? RB. Retinoblastoma. What is the emergency break? P53, right? Which gene is activated every time? CDK, cyclin dependent kinase gene, right? So this is all about G1 phase, right? See you in S phase. अच्छा सा क्वेश्चन इन हो क्या उस शाबे? So G1 phase